What's up, folks? We are going to do a video on the urinary system here. So let's go ahead and get to that. So the urinary system has several major organs, including the kidneys. See a kidney right here? Kidneys, of course, are going to make the urine. And then we've got the ureters. There's one ureter right there and another ureter over there. Ureters are going to transport the urine from the kidney, where it's made, to the urinary bladder. There's her urinary bladder right there. Urinary bladder stores it until it's convenient to release. And then our exit pathway is the urethra. So these are the major organs of the urinary system. So we're going to go through all of them, primarily talking about the kidneys. Now, overall, big functions of the urinary system. Okay? Big functions of the kidney. So urinary system functions and kidney functions are going to go hand in hand. One of them, our first one here, is the filtration and removal of waste from the plasma. That is number one. Now, one of the things we remove, in order to remove waste, we have to remove water. And by removing water, what we are going to regulate is blood volume. So when you make urine, you're making urine from the blood, and it's a liquid. So we are taking water out of our blood and turning into urine. And by doing so, we're regulating blood volume and we're regulating blood pressure. Lastly, we're going to regulate the osmolarity, or the concentration of the blood. So by adjusting how concentrated the urine is, we can adjust how concentrated the blood is. So basically, think about it this way. We're regulating the blood by putting stuff in the urine to get rid of it. All right, other functions of the kidney of the urinary system. We are going to make renin. Remember, renin makes you raise your blood pressure. We are going to make EPO, which of course is erythropoietin, and that's going to promote red blood cell synthesis. We're going to regulate body pH. If our body is too acidic, we make a more acidic urine. If our body is less acidic, we make a more basic urine. There's also a hormone made by your kidneys called calcitriol. And with a beginning with that calc, you can probably guess that it is going to play a role in calcium homeostasis. Lastly, the kidney does something called gluconeogenesis, where we make glucose from amino acids and fats. Okay, fantastic. Those are our big kidney functions. And before we dive into the kidney anatomy, we got to talk about the kidney's neighbors. We've met these guys before. Neighbors of the kidneys are the adrenal glands. And we got an adrenal gland here, and we got an adrenal gland here. Adrenal glands are also called the suprarenal glands. Suprarenal, because they are on top of the kidney. Oh, that's some beautiful penmanship right there. So the adrenal glands are going to secrete some hormones, aldosterone, which is one we'll talk about in this urinary system lecture, plus cortisol, which is our stress hormone, plus our two sympathetic fight or flight hormones, epinephrine or epinephrine. And there are other hormones as well, some of which we met in that very first lab so long ago. All right, so adrenal glands, we've met them. We'll come back to them a little bit later. But let's talk about some kidney anatomy. Yeah. So the kidney is very kidney-shaped. It's got a convex lateral surface. It's got a concave medial surface. There is a hilum where things are going in and out. We see a renal artery going in, a renal vein going out, and a ureter going out as well. Now, the kidney is surrounded by a layer of dense connective tissue called the fibrous capsule. So fibrous capsule is outside of the kidney right here. And then when we go into the interior of the kidney, there is going to be an outer region. So basically, like right here, this outer region is called the renal cortex. The renal cortex. And then we have these triangles. I have 1, 2, 3, 4, 5, 6 visible triangles here. There could, of course, be more. These triangles, each of them is called a pyramid. And overall, all the pyramids make up the renal medulla. Now, in between the pyramids, we're going to have these areas that look kind of like the cortex, and I'm putting some blue dots on them. And these areas in between the pyramids are called the columns. So renal columns are in between the pyramids. The pyramids make up the medulla. Outside the medulla, we've got the cortex. Now, urine is going to be made in the cortex. Urine is always going to be traveling through tubes in the pyramids. And the tip of each pyramid, let's put a green dot on the tips of the pyramids. The tip of each pyramid is known as a renal papilla. And urine always drips out of the renal papilla. The urine, when it drips out of the renal papilla, it ends up in a little tube, such as this one. Little tube called a minor calyx. There we go. So a minor calyx. And here I'm going to put purple in 3 minor calyces. When minor calyces join together, we are then going to get a major calyx. So minor calyces are tubes that carry urine from the pyramids. Minor calyces unite to make a major calyx. Eventually, all of the minor and major calyces join together right around here. Let's put a little star right here. All the minors and majors join together to make a kind of wide tube called the renal pelvis. The renal pelvis then leaves the hilum next to the artery and the vein, and it narrows and becomes the ureter. So that, my friends, is a little bit of basic kidney anatomy. And what we can do now also is do a basic pathway of urine flow. So we said urine is going to be formed in the renal cortex, travel through those pyramids, drip out of the pyramids, and it drips out of the pyramids from these the tips of the pyramids, which were called the papillae. And we end up in minor calyces, major calyces, renal pelvis, then the ureter, and like we mentioned in the very first slide, from the ureter down to the urinary bladder, and then to the urethra. Okay, fantastic. So we got some basic kidney anatomy, and we got some basic urine flow. Now, not only do we have to know the pathway of urine flow, we also have to know the pathway of blood flow in our kidney. So we're going to start with the renal artery. The renal artery, of course, comes off of the aorta. Absolutely. So the renal artery is branching right here, and we get several segmental arteries. So from renal artery to segmental arteries. We see some segmental arteries here. A segmental artery is going to branch and give us these arteries that go up in between the pyramids. These arteries going in between the pyramids are known as interlobar arteries. Then we get some arteries that are going to kind of arch over the base of the pyramids. So each pyramid at its base, at its wide cortical part, is going to have an artery that curves over it. That's called the arcuate artery. Then from the arcuate artery, look at these little branches going up into the cortex. These little guys coming off the arcuate arteries, they are known as interlobular arteries. Now, we're going to kind of pause here for a second. These are all of the big, and I'm making air quotes in the air here that you can't see, but these are all the big arteries of the kidney. We're going to do the microscopic arterioles and the capillaries next. But these are the big arteries. And these big arteries get us into the cortex. Why into the cortex? Because that is where we are going to make urine. Now, from the cortex, we're going to have some big veins to leave. So we got big arteries getting into the cortex. We're going to have some big veins to leave. Now, it's rather similar to the arterial path. In draining the cortex, we have interlobular veins. All these blue guys kind of spoking through the cortex, those are interlobular veins. Then at the base of each pyramid, the wide part of each pyramid, we're going to have an arcuate vein. Arcuate veins then have veins that are going down in between the pyramids, so in the columns, and those are called interlobar veins. The interlobar veins, so here I've got two interlobar veins, here's a third one. They're uniting right here. All the interlobar veins uniting. They're still called interlobar veins all the way until right here when we get back to the renal vein. So notice there is no segmental vein. There's a segmental artery, but there is no segmental vein. Now, this is our basic pathway into the cortex and out of the cortex. And we're going in and out of the cortex because the cortex is where we are going to make urine. All right, now let's zoom into the cortex and look at some of the microscopic structures. So we're going to zoom in on the cortex. Okay, here is an arcuate artery right there. There's an arcuate artery. Coming off the arcuate artery, we have got an interlobular artery, which is going up into our cortex. So interlobular artery, this red guy right here. Now the interlobular artery is going to give off a lot of little arteries. Each is called an afferent arteriole. Afferent tells me it's going somewhere. Well, where is it going? It's going to this ball of capillaries called the glomerulus. So this glomerulus is a ball of capillaries, and this glomerulus, this ball of capillaries, is super important. It is the place where we are going to filter the blood. It is where we are going to do blood filtration. It is the first step of urine formation. Each of your kidneys is going to probably have about 1 million of these glomeruli in the cortex. So each kidney has about 1 million of these glomeruli. Each glomerulus is a ball of capillaries where we are going to filter the blood, which is the first step in making urine. Now, once we filter the blood, the filtered fluid ends up going through all of this set of tubes here, these tan tubes. Eventually get into this big one. Eventually that filtered fluid becomes urine. As the filtered fluid becomes urine, we still mess with it. We do some stuff to it. And let's go back to our blood for a second. We have an afferent arteriole going into the glomerulus. We have an efferent or efferent arteriole coming out. Of course, since we're filtering the blood in the glomerulus, there would be a greater volume, greater number of wastes, greater number of ions, greater number of stuff in that afferent compared to that efferent arteriole. Now, from the efferent arteriole, there's two possible sets of capillaries we can get. There's a set of capillaries that are called peritubular. I see them up here, labeled nicely. They're around those tubes that the filtered fluid was flowing through. And their presence there is going to let us modify that filtered fluid. There's another group of capillaries down here called the vasa recta, the blood vessels down here. They also let us modify that filtered fluid, helping us turn it into urine. Eventually, the peritubular capillaries, the vasa recta, empty into the interlobular vein. Interlobular vein is going to go back to the arcuate vein and then back to our big blood vessels that we met before. All right, so now we've got not only the big blood vessels, but also the microscopic blood vessels of our kidney. We got a basic pathway of blood flow, which is fantastic. Okay, if we kind of go back to those microscopic blood vessels, I want to sum up a few things. So this picture here is showing me my microscopic blood vessels, but also showing me my little tubes in my kidney where I carry the filtered fluid that becomes urine. Now, okay, this is my afferent arteriole. It would come off an interlobular artery. It leads me to this ball of capillaries called a glomerulus. Now, that ball of capillaries is a site of filtration. We're going to filter the blood. We're going to make a liquid. The liquid actually has a name. The liquid that we make when we filter blood is appropriately called, what a great name, it is called the filtrate, because it just got filtered. The filtrate is going to flow through these tubes. They're named in this picture, but we won't worry about their name just yet. But the filtrate flows through these tubes, eventually becoming urine. Now, once blood filtration is done, we're not losing all the liquid, all the stuff in the blood. Maybe like 20% of the liquid and the solutes in the plasma becomes filtrate. The other 80% keeps going into the efferent arteriole. Oh, I want you to notice something. Look at the size of the afferent versus the size of the efferent. Afferent is definitely bigger. What this does is it enhances the pressure here to drive filtration, to force liquid and solutes out of the bloodstream. Now, the blood of the efferent arteriole goes up into these peritubular capillaries, or perhaps into the vasa recta, and the blood in the peritubular capillaries and the vasa recta, by flowing alongside the little tubes where the filtrate was, means we can exchange stuff between the filtrate and the blood. We can reclaim good things, that's called reabsorption. We can get rid of extra bad things, that's called secretion. And by the time we're done with filtering the blood, reabsorbing good stuff from the filtrate, adding any bad stuff to the filtrate, by the time we're done with that, we have made urine. So this is the basic process of urine formation, which is occurring in the renal cortex. Fantastic. All right. And here's a key factor here. If I am regulating the amount of water that I'm going to reabsorb, I can regulate blood volume. If I regulate how much water reabsorption I'm going to do, I'm going to regulate blood volume. The more water I reabsorb, so reabsorbing is going from the filtrate, so filtrate would be in a little renal tubule, and then plasma would be in my peritubular capillary. Reabsorption is going back this way. And if I regulate how much water I reabsorb, that means I'm going to regulate how much urine I produce and thus regulate my blood volume. Now, this picture over here shows me secretion, where I'm taking stuff from the plasma and adding it to the filtrate. I usually do that with wastes. Now, if I'm going to regulate the levels of chemicals that I either reabsorb or I secrete, I can regulate things in my blood like its electrolyte, its electrolyte concentrations, its calcium, potassium, sodium, etc., and also its pH. If blood pH was too low, then I would want to I would want to secrete hydrogen. If blood pH was too high, I'd want to reabsorb hydrogen. So let's talk about something. Let's recap for a second here. We filter the blood. We end up with this liquid called filtrate that flows through these little tubes. Eventually it becomes urine. But as it flows through these little tubes, it flows alongside blood. And I'm going to reclaim good stuff, stuff that I need, from the filtrate, put it back in the blood. That's called reabsorption. I'm going to add bad stuff or stuff that I don't need from the blood to the filtrate. That's called secretion. By the time I'm done with all three of these, we've made urine. And I can regulate what I do with these guys. And by regulating them, I can regulate things like potassium, sodium, calcium, hydrogen, i.e. pH, in my blood. And I can also literally regulate how much blood I have, which regulates my blood volume, my blood pressure. It is an ingenious and amazing system. Okay, so we were just talking about the process of urine formation. There's a glomerulus right there. Do you see that red thing in there? That's a glomerulus. Let's put a lovely arrow on it. There's a glomerulus right, boom, there. Here's our interlobular artery right there. Afferent arteriole, efferent arteriole, and arcuate artery right there. Just to get you situated. If you need to go back and review those blood vessels, now is a fantastic time to do that. Well, we were talking about the movement of blood, filtration, the movement of that filtered fluid, and the production of urine. Now, the set of all the things involved in that production of urine is collectively called a nephron. So the nephron is this group of structures in the kidney where I do three things. I'm going to do filtration, I'm going to do reabsorption, and I'm going to do secretion. And collectively, that means in the nephron, I'm doing urine formation. Okay, nephro means kidney. A kidney doctor is a nephrologist. So epinephrine from on top of the kidney. All good, similar root words there. So the nephron is just a name for a thing in the kidney. It's where I do urine formation. You have about 1 million nephrons per kidney. The first part of the nephron is the glomerulus. The glomerulus. And we saw the glomerulus hiding right in there. Not really hiding, but it's inside this structure called the glomerular capsule. So remember, the glomerulus is where we filter blood. The glomerular capsule catches the filtered blood. And then that filtered blood flows through something called the proximal convoluted tubule, which I am putting some blue dots on right now. Proximal means nearby. It's near the glomerular capsule. Convoluted means twisty. It is twisty. We'll talk about that twistiness in a second. And then we get something called the loop of Henle, also known as the nephron loop. The nephron loop. And the nephron loop, I'm going to put some green dots in it right here. The nephron loop goes down and goes up. We'll talk about it. And then we get another farther away twisty guy. Purple dots in it. That guy is called the distal convoluted tubule. And by the time we're done with all of these 5 structures, we've taken care of those 3 events: filtration, reabsorption, and secretion. And so we've made urine. So by the time we're done, by the time we get out of right here, we now have urine. We had plasma, filtrate, and by the time we get over here, urine. And this big tube that we see is going to get a bunch of nephrons emptying into it. These are all ends of nephrons here. And this big guy is called a collecting duct. It collects the urine from the nephrons. It travels down the pyramids. And the collecting ducts eventually merge together, get bigger and bigger. And collecting ducts empty at the renal papillae. Okay, fantastic. Let's go through all of these guys one at a time. Here's just another version of the same stuff. Before we go through them one at a time, there's our interlobular artery, afferent arteriole, taking blood to the glomerulus where filtration happens. Filtrate then moves into the glomerular capsule, the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, and the collecting duct. And meanwhile, recall that blood from the glomerulus goes into an efferent arteriole, and then up here we got some paratubular capillaries around the proximal and distal convoluted tubules. And down here we got our vasa recta, and then emptying into our interlobular vein. And within this structure, these tubes, these blood vessels, we do filtration, we do reabsorption, we do secretion, and we are making a urine. Okay, fantastic. Let's talk glomerulus. No, let's not talk glomerulus. Let's talk glomerular capsule. The glomerulus, of course, was the ball of capillaries where filtration occurs. The glomerulus is enclosed by a double-layered structure called the glomerular capsule. Double layer, 1, 2. Sometimes the glomerular capsule is known as the Bowman's capsule, old-fashioned name right there. And the glomerular capsule is going to receive that filtered blood. Okay. So if we talk about the two layers of the glomerular capsule, there is an inner layer, so a layer directly on the glomerular capillaries. And I see these cells here. See these guys? I'm putting dots on them. These guys collectively make up my visceral glomerular capsule. These cells are called podocytes. They're called podocytes because, check it out, they got all these little feet on them. Let's put some yellowish dots on their feet. They have all these feet on them. Notice how the feet kind of interlock with one another, leaving only a small space through which small stuff can go. And so, my podocytes, in other words, my visceral layer of my glomerular capsule, is very much involved in the filtration of blood. Alright, so that's my visceral glomerular capsule. Helps me filter my blood. There is also a parietal glomerular capsule. We see that out here. Check that simple squamous epithelium out. That simple squamous epithelium doesn't play any role in filtration, but it is going to hold the filtrate. So the filtrate ends up in this lumen of my glomerular capsule, and the filtrate then has nowhere to flow but onward into my proximal convoluted tubule, which got to cut off right there. So my parietal glomerular capsule doesn't do any filtration, but it plays a role in helping contain and move the filtrate. All right, excellent. Here we see the actual histological slide. This thing right here, I've actually got two things right here. This circle, I just drew a terrible circle on that. I'm going to get rid of that. That's an ugly circle. This circle right here, this is a little bit better. Still kind of ugly. There are two things here, two things in this green area. There is the glomerulus. So there are capillaries in here, and we can kind of see like the lumens of the capillaries. Dun, dun, dun. But then also I have my visceral glomerular capsule. In other words, I also have my podocytes. Now, what I can see, not only, in addition to these things, I also have this guy right here. Let's make an actual arrow, beautiful arrow. This guy, this is a layer of simple squamous epithelium, this blue thing. And so that layer of simple squamous epithelium is actually my parietal glomerular capsule. And by the way, I do see a lot of tubes here as well. The majority of these tubes that I'm seeing here are my proximal convoluted tubules. So Always cool to look at the real histology, in my opinion. You guys agree with me, I am sure. Let's keep going. Let's talk about the rest of these nephron structures. Coming out of the glomerular capsule, we have the proximal convoluted tubule, which I have put some dashed lines around. It is proximal because it's near the glomerular capsule. It is convoluted because it's twisty. So it gets the filtrate from the glomerular capsule. And the proximal convoluted tubule is where I do the vast majority, the lion's share of my reabsorption and secretion. I do most of my, not even most, like most of my reabsorption secretion in my proximal convoluted tubule. Now, because I'm doing reabsorption secretion there, I am going to need a couple of things. I am going to need a high surface area. Surface area is achieved by the twistiness plus the simple cuboidal epithelial cells that make up this tube. They have microvilli projecting into the lumen of the tube. That gives me more surface area. More surface area for reabsorption and for secretion. Now, the twistiness also helps give me enough time. When you're on a twisty road, it takes you longer to drive through it. So I get more time for reabsorption secretion as well. Plus, reabsorption and secretion are energy dependent processes. So I am going to need a lot of mitochondria. Look at all the mitochondria in these epithelial cells. Okay, that's our proximal convoluted tubule. From it, the filtrate travels through the nephron loop or loop of Henle, which gets involved in this complicated process, which we are going to just kind of sum up and say the loop of Henle helps us reabsorb water later on from our collecting duct. So later on, especially if you are dehydrated, you need to reabsorb water from your collecting duct. So you pee away less water. If you're dehydrated, you want to keep water, right? You want to pee away less, so you want to reabsorb more. The loop of Henle is going to help set the stage for water reabsorption in the collecting duct. It's a complicated process. There's something called the countercurrent exchange and countercurrent multiplier effect, and we're going to just know the end result. Forgive me if you wanted to know more. Okay, moving right along, we've got our distal convoluted tubule. Distal because it's farther away from the glomerular capsule. Convoluted because it's twisty. Gets the filtrate from that nephron loop, the loop of Henle. Now, the distal convoluted tubule is where we do reabsorption secretion, but we do it here in an adjustment way. It's where we're like, okay, what do we have enough of? What do we not have enough of? What do we need to keep? What do we need to get rid of? And there's hormones that are going to tell the distal convoluted tubule, hey, reabsorb more calcium, or hey, secrete more potassium. So it's where we make our homeostatic adjustments. Proximal convoluted tubule is where we're just getting back all the super important stuff. We're not really regulating it there, but in the distal we do the regulation. So we actually end up doing less reabsorption secretion in our distal, so it's often less twisty, fewer microvilli, fewer mitochondria. Okay, amazing. We leave the nephron, end up into the collecting duct. The collecting duct collects urine from several nephrons, maybe like a dozen or so. The collecting duct goes down. It's mostly simple cuboidal and columnar epithelium. Eventually, collecting ducts are going to get bigger and bigger as they merge, and they end at the renal papillae, emptying into a minor calyx. Now, we do perform some water reabsorption in our collecting duct. It's hormonally dependent. We've actually met the hormone that it depends upon. You know this hormone. I'll abbreviate it for you. You just shout out what it stands for. Shout out loud enough so your family members, whoever is nearby, if you're at work, shout it out at work, what ADH stands for. So ADH helps us reabsorb water from our collecting duct. Remember, the loop of Henle was necessary to set up the mechanics of water reabsorption in the collecting duct as well. All right. Yes, we are moving through our anatomy of kidney structures here. Let's go back to that first blood vessel, the glomerulus. We've learned a lot about it. We know it is a ball of capillaries. We know we have about 1 million of them in each kidney. We know it is the site of filtration. We have an afferent and efferent arteriole associated with it. Afferent going in, efferent going out. Notice how big the afferent is compared to the E. So efferent brings blood into my glomerulus, efferent takes blood away. This size difference means it's easy to get in, harder to get out. That raises the pressure in here, and that high pressure drives filtration. Now, notice the little podocyte sitting on top of my glomerular capillaries, helping me with filtration. Notice my parietal glomerular capsule right here. Notice my proximal convoluted tubule with its beautiful microvilli right there. All right. So there's our glomerulus. If we were to move onward from the glomerulus to our next set of capillaries, so we go into the glomerulus of the afferent arteriole. We come out with the efferent arteriole. We end up in these capillaries around the tubules, so they're called peritubular capillaries. They are going to work with my proximal and distal convoluted tubules to do reabsorption and secretion. Now, the blood pressure in my peritubular capillaries is really low. The osmotic pressure is really high. Both of these factors are going to facilitate the reabsorption of water. By the way, you are filtering, you are making probably over 150 liters of filtrate a day. Obviously, you don't pee 150 liters of pee a day. So you got to reabsorb like 99% of that water. And that water reabsorption is going to be helped by the low blood pressure, high osmotic pressure in your peritubular capillaries. All right, there's also the set of capillaries around the nephron loop, or loop of Henle. Those guys are called the vasorecta. Somebody thought they looked like straight vessels under the scope. And those guys are going to help me do water reabsorption if I need to, if I'm dehydrated in my collecting duct. All right, yes, we have now gone over all the anatomical structures, macro and microscopic, that we need to talk about for urine formation. That being said, I feel like this is a awesome spot to stop this video. Urinary system video number 1 is officially done. I'll see you for urinary system video number 2.